Hello everyone, my name is Jiangnan. I'm a student from Zhejiang University in Hangzhou, China. I am so honored to have this opportunity to communicate academic questions and ideas. The topic of my presentation is a bio-inspired olfactory sensor based on organoid on a chip. My presentation will include these three parts. First, the background of our research, second, experiments and results, and the last, some conclusions we have got. Neurodegenerative diseases are gradually becoming the main burden of society. There are numerous studies in this area. However, the etiology and pathogenesis are so complicated that have not been fully elucidated. For most neurodegenerative diseases, there are no effective treatments. Studies have found that brain regions related to olfactory function are closely associated with cognitive decline. Olfactory dysfunction is an early symptom of some highly prevalent neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, and so on. Although the pathogenesis remains inconclusive, as a prominent clinical feature that predicts the subsequent development of neurodegenerative diseases, research on olfactory dysfunction is of great significance. The mammalian olfactory system is considered to be one of the most efficient chemosensing systems due to its extraordinary ability in odor detection. The olfactory system consists of three parts: the main olfactory epithelium, olfactory bulb, and olfactory cortex. There are a lot of olfactory sensory neurons in the olfactory epithelium, which are bipolar transduction cells that transmit olfactory information to the brain. Individual olfactory sensory neurons express one specific odor receptor. Once the odorant is bound to the odor receptor, the receptor activates the attached G protein on the inside of the olfactory receptor neuron, and the odor information will be transmitted to the olfactory bulb as action potential. Organoids are novel three-dimensional model proliferated and cell-assembled from stem cells. Organoids exhibit similar structure and function with human organs, working as biomimetic models for biological research. In this study, three-dimensional culture technique was used to induce mammalian olfactory epithelial stem cells to differentiate into olfactory organoids, which has the same cell type with olfactory epithelium. Mesh is used to create a better three-dimensional environment in the culture of organoids. With the addition of related growth factors, stem cells will proliferate and differentiate to organoids. This slide shows the culture process and the image of olfactory organoids. After about 2 weeks of culture, the olfactory sensory neurons mature gradually, which can be defined by their expression of the olfactory marker protein. This slide shows the confocal images of olfactory organoids immunostained with antibodies against tubulin and olfactory marker protein. We observed the presence of tubulin positive cells, indicating the generation of neuronal cell lineages in organoids. In addition, this image shows that several cells also express the olfactory marker protein, demonstrating that mature olfactory sensory neurons were generated in organoids. This is an essential condition for organoids to be used as a sensitive element in later experiments. Furthermore, calcium imaging was carried out to verify the function of the olfactory organoids. Organoids were collected and reseeded onto the confocal dish and then cultured for 24 hours. After calcium downloading, imaging was carried out using confocal microscope. Odorant solution was applied by bath application. Acquired sequences of images were converted to the ratio of delta F and F, where delta F was the real-time fluorescent intensity change relative to F, and F was the average baseline fluorescence values before odorant onset. Cells in organoids of interest were labeled with a fixed area of region of interest using ImageJ software, and fluorescence change over time in this region was calculated. This figure shows that cells in organoids responded to odorant mixture of high concentration. With the support of the results mentioned above, We combine the organoids with sensor chip to monitor the physiological activity of olfactory organoids. Biosensor is a kind of analysis systems using cell, tissue, and other biomaterials as sensitive elements to convert chemical signal into electrical signal. It can achieve label-free real-time and long-term monitoring of cells and has been widely researched and used as a tool for medical, environment, food, and pharmaceutical field. The development of bio-inspired olfactory biosensing technology makes it possible to mimic biological olfactory system. To overcome the limitation of olfactory biosensor in sensitivity and specificity, we present a novel olfactory biosensor based on three-dimensional organoid on a chip. Organoid on a chip is a development of organ on a chip, which combines organoids, sensor chip, and a microfluidic system. It is a system that can monitor the physiological activity of organoids quantitatively and sensitively in real time, and can model the functional units of organoids in vitro. This technology has important application prospects in pathological study and drug discovery of disease. A multi-channel microelectrode array chip was designed and processed to acquire and record the odor response signals of olfactory organoids. When the organoids are transferred and fixed to the sensor chip and given an odor stimulus, the corresponding signals of neural activity are recorded. The feature extraction and pattern recognition of the signal are completed by the neural decoding algorithm, and then the odor recognition model is constructed to realize the hand sensitivity, specificity, stability, and rapid detection and recognition of the odor. This slide shows the signal of neural activity before and after the odor stimulation. Spikes that represent high-frequency neuronal excitations were recorded, and the fire rate of the spike chain increased obviously after the addition of odorant. And finally, this slide shows the conclusions we have got. Olfactory organoids express tubulin and olfactory marker protein, indicating the generation of neuronal cells and mature olfactory sensory neurons. Cells in organoids respond to odorant mixture of high concentration. The fire rate of the spike chain increased obviously after the stimulation. This olfactory biosensor based on organoid on a chip provides a more accurate and efficient platform for detection of odor. That's all for my presentation. If you have any question, please feel free to contact me. Here is my email address. That's all. Thank you.